Modern biology is standing at an intersection. We are moving past the era of simply describing living systems and entering a phase where we must systematically predict them. This video shows the reality of that transition. Thousands of disconnected data points and raw biological sequences are flooding modern computational research systems every second. The primary bottleneck for researchers today is no lack of data or shortage of software. The true constraint is the complete absence of a cohesive modeling framework to organize the noise. There is an increasing reliance on artificial intelligence and routine Python code generation to handle this volume. Executing syntax, however, is not the same as understanding computational biology. When we rely purely on software without a preceding mental model, we compromise the scientific validity of our research. The tool becomes a black box, and the output becomes untestable. Raw data processed without a strict mental framework is highly organized noise. The scientific challenge we face is conceptual, not computational. Systemic modeling is the deliberate art of intelligent simplification. To achieve this simplification, the modern biologist must adopt a dual role, operating as part philosopher and part engineer. As philosophers, we must ask strict structural questions about how a living system physically operates and what specific variables drive those operations. As engineers, we must possess a deep understanding of how computational tools are structured and executed to test those exact variables. This diagram shows the exact intersection of these two domains. We call this the abstraction level, and it is the specific space where true computational science happens. Mastering this level allows a researcher to prune away irrelevant biological variables and isolate the core mechanism long before writing a single line of code. The ability to determine this correct level of abstraction is the precise characteristic that separates a true scientist from a mere programmer. The first step in building this mental algorithm is to evaluate life at its most granular foundation, the molecular and enzymatic scale. Here we see a dynamic biochemical reaction pathway. At this scale, we are observing the non-stop physical interaction of molecular structures. By applying our abstraction framework here, we can model reaction kinetics and accurately map highly nonlinear biological behaviors. From there, we move our focus one step higher, entering the genetic and cellular level. This animation illustrates gene regulatory networks, the architecture driving cellular decision-making. Notice how edges between nodes function like binary switches, dictating specific responses. Before collecting data, we must translate raw hypotheses into these transparent switches. At the micro level, life is entirely governed by mathematical kinetics and logical switches that we can isolate, map, and predict. We then expand the framework to a third scale, the macro view of metabolism and systemic biological networks. Our focus shifts from analyzing single, isolated pathways to measuring massive data flows and assessing metabolic flux balance. This chart shows data flowing through a metabolic network. The system is in constant motion, continuously seeking balance and optimization across thousands of interactions. The central complexity at this scale is emergence, the phenomenon where simple, isolated parts combine to create highly unpredictable macro behaviors. Standard coding practices fail to capture this emergence if the underlying structural philosophy is flawed. However, a strong mental algorithm isolates these specific emergent patterns directly from the overwhelming background noise of the metabolism. Mapping these precise behaviors allows us to accurately predict how the whole system will respond to external stimuli. True systemic prediction requires zooming out far enough to let these emergent properties reveal themselves mathematically. Finally, we arrive at the overarching scale of living systems, macroscopic ecological networks, and the biosphere. This visual represents a dense, dynamic ecosystem, focusing entirely on the continuous physical motion of interacting populations across a geographic space. The exact same logic of nodes and edges applies here. The only difference is that the nodes now represent entire biological populations instead of single genes. This matrix shows how applying our level of abstraction allows us to model complex population dynamics and test the structural stability of the entire biosphere. The mental algorithm of abstraction is universal. It scales perfectly from mapping a single enzyme to calculating global ecological stability. We must now shift our focus from the theoretical scales of life to the operational reality of scientific publishing and research output. Adopting this specific mental algorithm translates massive biological complexity into testable, publishable hypotheses. An abstract systemic model directly forms the foundation of the results section in leading scientific journals. Forming this mental model is the absolute prerequisite. It must be finalized before touching any heavy datasets or writing complex Python code. To master systemic thinking, we are hosting a specialized 2-hour webinar titled From Molecule to Biosphere. The session takes place Wednesday, Mordot 14th at 7 p.m. Participants will receive full access to the recorded video, source code files, and Python templates. In the artificial intelligence era, computational tools are infinite, but engineering a precise biological model remains the ultimate scientific currency.